What I'll cover next are some clinical pharmacologist considerations when developing oligonucleotides. And the intent here is just to give a couple of an overview and a few examples, of important differences or important studies that we should consider in the development of this type of molecules. As Suzanne covered initially with her talk, there are different classes of oligonucleotides that are being developed as therapeutic agents. Because these molecules play a significant role in regulating the biological process with the intent of treat the disease, they end up having a very prolonged pharmacological effect. All these unique characteristics of oligonucleotides and the limited knowledge that we have when it comes to clinical data, PK, safety, and efficacy, then makes the development of this type of molecules more challenging. I think we are evolving in the clinical pharmacology field in terms of how much we understand for this type of compounds, and we are building sort of a database, if you wish, that can help us guide the development of any new molecule that comes from this class, from making chemical modifications or conjugations to treat different disease. So when developing oligonucleotides, I think it's important that you consider all aspects of clinical pharmacology, that you understand the PK properties of your drug and what can impact exposures to better define and optimize the doses for the patients. What I'll cover next in this presentation is essentially go over a bit of the pharmacokinetics properties with some examples, the drug-drug interaction data, and then renal and hepatic impairment. As Martin covered during his presentation, the PK of oligonucleotides are primarily a function of their backbone and chemistry. Modifications to that backbone and conjugations to it have resulted in changes in tissue binding, protein binding, and bioavailability or half-life. One example of that is presented in the figure on the right, where you see the top three curves are the profile of a parent ASO, and the bottom three curves is the profile of the same ASO now with a GalNAc conjugation. As Martin explained, certain conjugations then target specific tissues and increase distribution into the tissue. And that results in then you having a lower plasma exposures, which is being demonstrated here in that figure. But overall, elimination half-life of these two agents is very similar and comparable, but there are differences, in the plasma exposures and the tissue distribution. These aspects are important to be investigated in a non-clinical setting, so you can look at the absorption, distribution, elimination, etc. And specifically when it comes to tissue distribution, those are important information to collect in the non-clinical space. There has been certain consistency in terms of the distribution across different tissues and across different animals that makes the extrapolation from the non-clinical information to the clinical setting a little bit easier. The reason why considering running or having this information in a non-clinical species is to support your clinical setting or what experiments you want to perform or your studies that you need to perform when going into humans. But also radiolabel studies with oligonucleotides are usually not feasible and not required because of that long tissue half-life that the compound has, and makes this type of studies really challenging or difficult. But that being said, these are still things you need to explore in your phase 1 studies. In terms of a clinical pharmacokinetics, oligonucleotides are usually dosed as IV or sub-Q, but there are efforts in developing oral formulations that would allow oligonucleotides to be taken orally and make it more comfortable for the patient. The clinical profiles are very comparable, similar to the non-clinical profile as Martin described. You see fast absorption after sub-Q administration. There seems to be a flip-flop kinetics with that plateauing of exposures until you see a clear elimination phase. In general, you see dose proportional pharmacokinetics at the clinical doses, although some non-clinical more than dose proportional exposures may be observed. If you see saturation, depending on the type of oligonucleotide you're working with, there may be saturation in the distribution into the tissues if your target specific receptors that in consequence will result in having more drug in the plasma. Because of the long-lasting pharmacological effects that you see with some of these drugs, the dosing is usually sparse within weeks or sometimes months, and therefore there's no PK accumulation that is seen. Demographic parameters like age, gender, race do not impact the PK properties of these compounds, although some correlation has been seen with body weight. And some of these agents are dosed in a mg per kg basis, while some other oligonucleotides are dosed as a flat dose in milligrams. The conjugation can impact the distribution of oligonucleotides, as Martin described or discussed previously. The plot you see here is an example from givosiran, which is a GalNAc conjugated ASO. The solid lines represent the parent GalNAc drug, and the dashed lines you see there are the main metabolite that were observed in plasma samples, which is formed based on the loss of one nucleotide from the 3' end of the molecule. In general, the parent is the major component or the main circulating species that you see in plasma, and these metabolites tend to be in a very smaller proportion or percentage relative to the parent. But in this case, the main metabolite for givosiran was actually present in a good amount, and it was about 35 to 75% of the parent across the different dose ranges that were investigated. So it is important that during the phase 1, you look into doing some investigation in terms of the parent, the presence of the metabolite, and how the magnitude of how much metabolite is present relative to parents. Some of these metabolites can be active and can contribute to the pharmacology. So understanding the amount of the presence is important in plasma, as well as looking at urine, because this molecules are excreted in urine. And then understanding the contribution of urine clearance is important to help you decide later on if a renal impairment study is necessary. In terms of drug-drug interactions, there is a low risk of drug-drug interactions with this class. Usually, oligonucleotides are metabolized by endo or exonucleases. There's no involvement of CYP450 enzymes in the metabolism of these drugs, and they're just, as I explained, the endonucleases would just cleave a nucleotide out of the ends of the oligonucleotide, the 3' or the 5' end, and make shorter length fragments, while the endonucleases may cleave the molecule in more inside in the middle of the structure. But because these are large molecules, negatively charged, hydrophilic, they do not typically interact with CYP enzymes. They have not been demonstrated to be substrates, inhibitors, or inducers. It's the same thing with transporters. That being said, there is a possibility of drug-drug interactions from a mechanistic perspective, which can be related to the pharmacological effects, and I have one example on that that I can present later. And there's also a possibility of indirect DDI due to cytokine storms. So knowing how your drug behaves in vivo is important for you to decide what studies you need. So you need to look on a case by case and make that case whether you need to run a DDI study or not. There is no specific regulatory guidance on DDI, so the examples are shown next. It's essentially applying the current DDI guidance to decide whether clinical DDI is needed or not. In most cases, the clinical DDI was investigated either in dedicated studies or via population PK analysis. For example, if there are common co-administered drugs in the patient population, you can explore drug-drug interactions with a common co-dose drugs in population PK approach. So far, from the drugs that have been approved, all of them included some DDI assessments in vitro or in vivo, but there has been no specific dose change or recommendation of any dose change due to drug-drug interaction. In general, I think the in vitro studies for DDI is very simple and straightforward to run, and there is recommended that you do collect that information to support your submission. From victim DDI perspective, there is little to no risk. I think many results or studies have demonstrated that these are not substrates of CYP450. There is a publication where they investigated in vitro DDI for different GalNAc sRNAs and did not show any involvement of CYPs in the metabolism of different type of compounds, which is presented in the table here on the slide. There may be a little bit of contribution of some of these enzymes, which is a very low percentage when it comes to just end the methylation or all the methylation of the link for the conjugated GalNAc sRNAs. But overall, no significant contribution. And in my experience, we actually helped one of these molecules with the submission for one of these molecules, which did not specifically include a CYP phenotyping assay, but we are able to actually build a case based on the knowledge of the metabolism of these drugs and based on the knowledge of similar oligonucleotide classes or conjugates, that the data was not needed. From a perpetrator DDI risk, there's also data suggesting that oligonucleotides are unlikely to have any perpetrator DDI effect. As I mentioned, the past approvals at the FDA of this class of molecules, they all included some in vitro perpetrator assessments, but there have been no in vitro induction or inhibition of CYP enzymes that has been reported that are of clinical significance. The same publication that investigated the different GalNAc conjugates also look at substrates, also look at them as inhibitor of the enzymes, and there are a couple of these compounds that actually had a weak inhibition of 2B6 and 2C8 as presented on the table here. The authors then suggested that the results of the in vitro DDI inhibition then be assessed in terms of the risk of a clinical DDI by just performing the assessments proposed or suggested in the FDA and the EMA guidances and looking at the in vitro KI inhibition relative to the in vivo Cmax. In this case, the maximum concentration was actually, they looked at the plasma as a first step, but they also considered the liver concentrations. Because now GalNAc conjugates, they target the liver, they tend to have liver concentrations that are multiple fold higher than the plasma. So similar assessment was performed using the predicted liver concentrations just based on the distribution of these molecules into the liver that was investigated in the non-clinical studies. Overall, despite of seeing some in vitro inhibition after all of the assessments, there was essentially no clinical DDI risk that was estimated for these molecules. As I mentioned, also sometimes these drugs are to be administered in addition to other treatments. So, for example, mipomersen is indicated for hyperlipidemia as an add-on therapy to statins. And then they looked at investigating potential for drug-drug interaction between mipomersen and statins, and also ezetimibe, which are the two treatments that patients with high lipid or LDL-C levels can be on continuously, and then you start a treatment with meformin as an add-on to help maintain the levels, the LDL-C levels low. Because these are continuous and common co-administered drugs, then investigating the DDI of these agents is a question that you likely received from the agency. Based on the in vitro data, there was no rationale to think of any potential DDI because meformin is not an in vitro inhibitor or does not interact with CYP450s. However, there's always the potential for a local PD effect as these drugs are acting in the liver and depend also on different biological mechanisms for the response and elimination, which can impact each other's drug or exposures. In the end, the overall conclusion here is that there was no impact of simvastatins or ezetimibe on the meformin exposures. However, meformin did reduce the Cmax that was observed for simvastatin and ezetimibe by about 50%, but that was not considered clinically relevant. Statins are drugs that have a high variability in the PK, and even if you look at the PK of patients, it can have really a wide range of concentrations observed. And at the end of the day, because of the long prolonged effect that we see with these agents, the assessments of PD response is important when you look at drug-drug interactions, where you may see a difference in exposures, but you may not see a difference in response. An interesting example is with givosiran. There was a drug-drug interaction with givosiran, which was not related to direct inhibition of the enzymes. So givosiran is an inhibitor of ALS1 mRNA, and ALS1 is the first and the rate-limiting enzyme in the heme biosynthesis. So if you decrease the hepatic heme, there is the reduction of the proteins that depend on heme, and that's the case for CYP450. So just based on the pharmacological cascade of events, there could be a reduction of proteins and impact then the CYP450 function. This was an interesting study from my perspective, a cocktail study that tested five major CYP450 enzymes in one study, and they investigated the impact of givosiran on 1A2, 2D6, 2C9, 3A4, and 2C19. The results demonstrate here on the figure on the forest plot that you have on the slides just show that there was a moderate inhibition of 1A2 and 2B6, and the increase in exposures of the sensitive substrates were in the 2 to 5-fold range when they were co-dosed with givosiran. A less moderate effect was observed with other enzymes, the 2C19 and the 3A, and no effect was observed with 2C9. The results of the study then had the label implications. So it's recommended that substrates that have a narrow therapeutic index or window for the 1A2 and 2D6 be administered with caution with givosiran. There has been no dose change. It's just a warning for this concomitant drugs. Switching gears to renal excretion now and contribution to the elimination of oligonucleotides. As Martin explained, there is accumulation of these drugs in the kidney, and they do undergo renal clearance. Usually GalNAc conjugated, they're less, you see less of a renal clearance for those type of molecules, but still it contributes to some part of the clearance of the parent. The approved drugs have investigated the impact in renal clearance mainly via a population PK analysis by enrolling patients that have different degrees of renal impairment into the clinical trials. and investigating the impact or differences in PK or exposures for the drug across these different populations. There was no dedicated renal impairment study in the submissions except for inclisiran. Inclisiran was approved early in the year in 2020 in Europe, and a dedicated renal impairment study was reported for that compound. Interesting enough, if you look at the results for the renal impairment for inclisiran, so inclisiran is also GalNAc siRNA. It's indicated for the treatment of high cholesterol and high lipid on top as an add-on therapy to statins. The figure on the left just showed the PK profile of inclisiran in different groups, in groups with different renal function. The yellow curve on top is the severe renal impairment group, and the green curve on the bottom is a normal renal function group. So there was a clear impact of renal function on inclisiran exposure, which demonstrated the significant contribution of renal to the elimination of this compound. However, if you look at the figure on the left, this is the LDL-C levels by different renal functions. Essentially, there was no significant difference in the PD response in terms of reduction of LDL-C in the plasma across the different renal impairment groups. So despite of the higher exposures, the PD response actually did not change with increase in the inclisiran exposures due to renal impairment. Investigations were also done in the clinical trials and looking at PD response as LDL-C reductions in patients that have different levels of renal function, and all of those demonstrated that there was no impact. So the end recommendation in the label was there was no dose adjustment necessary for patients with mild, moderate, or severe renal impairment and end-stage renal disease. From a oh, sorry, it's just from a very quick, as I understand we're on the top of the hour, from a hepatic impairment perspective also, there has been no dedicated hepatic impairment study in a publication that reviewed the submissions for 5 different oligonucleotides. Some of this did do assessment of hepatic impairment impact on PK, doing population PK analysis. Again, for inclisiran, there was a dedicated hepatic impairment study. There was an impact of hepatic function on exposures, and there was an increase of about 2-fold in PK with increase in the degree of renal impairment, but the results on the PK and exposures did not impact the PD response. And the consequence in the label is that there was no dose adjustment that was required for inclisiran in hepatic impairment, despite of the differences of PK that were observed. So overall, in terms of the clinical pharmacology aspect, I just wanted to share a couple of examples and results of different studies and approaches that have been applied to the development of oligonucleotides. It is important that we leverage the knowledge that is available for the class, as well as the clinical pharmacology knowledge on how we do drug development to support understanding how intrinsic, extrinsic factors impact PK and what is the optimal dose regimen for patients. Regulatory agents are open and willing to work with the sponsors in developing oligonucleotides, so engage with them early. They have guidances on how to engage with them in that aspect. So I think it's important to start a conversation as soon as you can. Key for this class, and maybe it's not usually not done for other molecules, is to also consider including PD assessments with your phase 1 studies, as changes in PK may not necessarily result in those adjustments just based on the PD response. And the FDA is also looking at publishing a draft guidance on clinical recommendations to support IND submissions of oligonucleotides. So there's more to come there. And with that, I just want to thank you for your attention. and we are going to open up for questions now.